students, welcome to the biology class. Today we are going to discuss very important topic from your same chapter, molecular basis of inheritance, and the name of that topic is transcription. What do you mean by transcription? Transcription means the synthesis of RNA from DNA. So the process of copying genetic information from one strand of DNA into RNA is termed as transcription. The principle of complementarity governs the process. In the RNA except adenine, now the base pair with the adenine is uracil instead of thymine. So we know in RNA the base pairs or the nitrogen bases which are present in the RNA, they are adenine, guanine, cytosine and uracil. And the base pair which is present in the DNA, that is adenine, guanine, cytosine and thymine, and the difference is of adenine, and so instead of thymine in RNA we are having the uracil. So unlike replication, only the single stranded fragment of DNA get copied into the RNA. So first we will discuss what do you mean by transcription unit. So what is transcription unit? Means you know the DNA is double stranded as it is being shown in this systematic structure of transcription unit. The one strand is from 3' to 5', and the another strand is from 5' to 3'. Means they are antiparallel to each other. DNA is a double stranded but RNA is single stranded. So only one strand will participate in the synthesis of single stranded RNA. And as we know, the polarity of the formation of new strand is from 5' to 3'. So the template which will participate in the synthesis of RNA is the strand from 3' to 5', and that strand is known as your template strand. Opposite to that strand, parallel to that strand which is not participating directly in the RNA synthesis, but that gives the information present on the RNA, that is known as the coding strand, or you can say as the sense strand. So what is the transcription unit? That is consists of three regions. So number one is the promoter, and it binds for the site of RNA polymerase for initiation of transcription. Number two is the structural gene, and number third is the terminator. And the enzymes which is playing the very important role in the synthesis of RNA is DNA dependent RNA polymerase. They helps in DNA replication by catalyzing the polymerization only one direction, that is 5' to 3'. The DNA strand that has a polarity 3' to 5', they act as a template strand, and it is being referred as the template strand. And opposite to that, which is not get transcribed or which is not being participating in the transcription, is known as the coding strand and has a polarity 5' to 3'. Its sequence is same as that of the RNA, except the T is replaced by the U in RNA. The promoter is located towards the 5' end, that is the upstream of the structural gene of the coding strand, and provide the binding site for the RNA polymerase. The sequence of the DNA located towards the end of 3', that is the downstream of the coding strand, where the process of transcription would end, is known as the terminator. Now you can see that is your promoter which is being attached to the 5' position of the coding, because the new strand polarity is also from 5' to 3'. So promoter means from where the transcription will initiate. Then we are having a structure genes in which we have a polymerization of ribonucleotides with the help of the enzyme, that is DNA dependent RNA polymerase, and at the end where the synthesis is going to be complete, terminate. So there is present another terminator, that is as a 3- end of a coding strand. So we are referring it as a coding strand because the polarity of the new strand is same as that of the coding strand. So this is about the transcription unit, and now we can have a transcription unit and the genes which are being participating. Very important, what is cistron? The segment of the DNA coding for the polypeptide is known as cistron. So this segment of the DNA which helps in the formation of RNA. From RNA the polypeptide or protein synthesis will take place. That segment of the DNA is known as the cistron. In eukaryotes, the transcription unit possesses the structural gene specific only for a single polypeptide, so that is monocistronic, whereas in the prokaryote, the transcription unit processing the structure genes for many polynucleotides with the same segment, so that is known as the polycistronic. In the genes in the eukaryotes, they are split into the coding or expressed of the DNA, that is known as the exons, and they are also having the non-expressible sequence of the DNA, that is known as the intron. But in the functional RNA, we are having no only exons, no introns. So how the transcription takes place? First in prokaryotic, that is in your bacteria. So in prokaryotic, the structure gene is polycistronic and continuous, because from one segment number of the polypeptides can be synthesized, and in bacteria or in the prokaryote, the transcription of all the three types of RNA, as we know we are having a three types of RNA, that is mRNA, tRNA, rRNA. They are taking with the help of a single dependent enzyme, that is known as RNA polymerase. All the three RNAs are needed to synthesize the protein in the cell. So mRNA, as the name indicates, that is a messenger RNA. They provide template, means they bring the message in the form of a codons, and they brings the amino acid, whereas the tRNA that brings the amino acid, read the genetic code, and tRNA they plays the structural and the catalytic role in the translation. The transcription is completed in 3 steps. So the first step is initiation, as we have discussed in the transcription unit, that is your promoter. So this is the segment of the DNA, means through which it is DNA helix, and the one particular segment which is going to be unwind, suppose this is the cistron, where the unwinding, where the breaking of the hydrogen bond takes place, and at the 5' position of the coding strand, the promoter, that is in the case of the prokaryote is sigma, is going to attach, and then the formation of a ribonucleotides with the help of the RNA polymerase, they get attached, they will get polymerized in the form of a structural gene at the end, where there is a terminator, that is the Rho in case of the prokaryotes, and when that is being terminated, they get separated from the DNA and takes place into the cytoplasm of the cell. So we are having the initiation, elongation, and termination. So one by one we are going to discuss that. Sigma factor that recognizes the start signal as a promoter, and the promoter region on the DNA, which along with RNA polymerase bind to the promoter to initiate the transcription. It uses, that is, nucleotriphosphate at substrate. It may be adenine, guanine, cytosine, and uracil, and polymerizes the template dependent fashion following the new complementary. Matlab A will go with U, and T will go with A, G with C, C with G. And next is your elongation. The RNA polymerase, after initiation of RNA transcription, loses the sigma factor continues the polymerization of ribonucleotide to form the RNA. It facilitates opening of the helix, continues the elongation with only the short stretch of RNA being bound to the enzyme at the time. So next is your termination. Once the RNA polymerase that reaches the termination region of DNA, the RNA polymerase is separated from DNA RNA hybrid. As a result, that is nascent means single stranded RNA separate. This process is called termination, which is facilitated by the factor that is Rho in prokaryotes. So in prokaryotes, mRNA, they are not requiring any processing because that is a functional. So both transcription and translation is in the cytosol, means in the cytoplasm. It can be said that transcription, translation, they are coupled together, as many times translation can begin much before the mRNA is fully transcribed. So in the prokaryotes, that is the structural gene is polycystic and continuous. Same enzyme, that is RNA polymerase, is used for the synthesis of mRNA, tRNA as well as rRNA. And we are having the initiation with the sigma factor at the 5' end of the coding strand, that is known as your promoter. Then there is the polymerization with the help of a RNA polymerase enzyme, and at the terminator end they are getting separated with the help of another terminator factor that is known as a RAHO, and that takes place in the cytoplasm of the cell. Now next we will discuss about the transcription in eukaryotes and how they are being different from the prokaryotes. The first is structural gene of the eukaryotes is monocistronic and discontinuous. The process of transcription that takes place in the nucleus instead of cytoplasm. Coding gene sequence that is called as an exon, they form the part of mRNA. Non-coding sequence, they are called as intron, and they are being removed during the processing of the RNA, and again the exons, they are going to be joined. So in the eukaryote, 3 types of enzymes are being used for 3 types of the RNAs, that is RNA polymerase 1 that transcribe the rRNA, then RNA polymerase 2 that transcribe the precursor of mRNA, that is the heterogeneous nuclear RNA, and RNA polymerase 3 that is transcribe tRNA, and these are the small subunits. So after the formation of RNA, same as that of the prokaryotes, with the help of the promoter, structural genes, with the help of the RNAs and the terminator in the nucleus, that is having the heterogeneous RNA, means non-functional, because the primary transcript, so the post-transcriptional modification in the eukaryotes is there. The primary transcripts are non-functional, containing both the coding region that is exon and the non-coding region that is intron. Such type of the RNA is known as the heterogeneous RNA, and to make it functional we have to go for the processing of mRNA, which is not in the prokaryotes, because in the prokaryotes they are not having any sequences of coding and non-coding. The whole of the transcript is functional. But in eukaryotes, they are having the for non-functional as well as the functional regions, that is coding region that is known as exon, and the non-coding region that is known as the intron. So for the processing, we are having the 2 additional processes. So they undergoes the slicing and 2 additional processing, that is capping and tailing. In capping, the unusual nucleotide, that is methyl guanosine triphosphate, is added at 5' end of heterogeneous RNA. And in tailing, so here you can see that is called as a capping, which is, this is your exons and introns, that is your heterogeneous RNA, and you have to make it functional. The first process is capping, the second is your tailing. So in tailing, the adenylate residues they are added at 3 dash end in the template dependent manner. And after having the, you can say, capping and the tailing, now they undergoes the slicing, and where the introns are removed and exons are joined to form the functional RNA and form the nucleus, they will enter into the cytoplasm, which will helps in the formation of your new polynucleotide, that is your protein in nature. Fine. So this is about the prokaryotic transcription as well as the eukaryotic transcription. Till then, thank you.